You guys have, is it? Look, it's 145, right? So by my watch, it's 147. But there's a big echo from the front here. I think you're okay in the back. I'm Wayne Koch. This is the panel on epigenetics and genetics in head and neck cancer. And you can see on the title slide the names of our 4 panel members. I'm going to just make a few brief introductory remarks. We're in this cavernous room, so those of you in the back who want to be able to sneak out, that's fine. If you're committed to the time and you want to move down front, I'll invite you to do that. The unifying theme of this group of talks is fairly broad. In this title, you can see that. And my concept, as I've tried to do what the meeting organizers asked in putting this together, is to keep it somewhat philosophically broad. So you'll hear us talking about some specific things that are going on in our own laboratories, but then we want to try to pull it back together and put it into a more global package and say, what does this mean? How do we make sense of the huge amount of information that's available to us with the now 15 to 20 years of molecular research that's been going on, and make it make sense as we move forward. So that's my goal. We'll take the... I personally have nothing to disclose. I think our panelists each should have this slide as well. So I thought I'd borrow a slide from my colleague, Joe Califano, which many of you have seen, just to begin the conversation. And that is that with the old Vogelstein kind of thought about tumor progression, we now continue to agree that cancer is a genetic disease brought about by a series of accumulated alterations in the gene coding mechanism. We've changed our thoughts from saying that this is due to mutation and deletion only and added some other epigenetic kinds of alterations that you'll hear about. But there still is this concept that we go from something simple to something increasingly complex in terms of the aberrations of the genetic coding and translational system that's going on. And as that accumulation of alterations occur, so also does a clinical progression from a little bit disordered to greatly disordered cells with cell function that follows. And so that's the concept that Joe put forth in this slide. Now, in my opinion, not all tumors go through the same genetic kinds of pathways, and in fact, therefore also the phenotypic changes that you see depicted on this slide with dysplasia and leukoplakia, and then more dysplasia and erythroplakia, and then invasive carcinoma, that gets skipped over in some cases, or at least apparently so that you have HPV-related disease that often presents first as a metastatic lymph node with a very tiny primary that might even involute and never show up. So clearly we're not dealing with just one concept, even as we look at what the translational ramifications of the genetic alterations are. So we're challenged now because our tools have gotten more and more powerful. And this is just a generic picture of a microarray chip. This could be a cDNA, it could be an siRNA, it could be all sorts of different microarrays. But the point is, we can get many, many, many data points about alterations within populations, and we've got the garbage in, garbage out problem still there, but we also have this huge bioinformatics problem. What do we do with thousands of data points? How do we make any sense of that? And how does that fit in with information about a specific gene, the sorts of things I've done with P53, where we're looking at 400 or 500 cases and asking about just the P53 spectrum of mutation. Somehow those things fit together or need to fit together in our minds. So that's one of the things that we'll hear about today. This is a slide from the Vogelstein Lab's recent Science article. Bert and his group, in a tour de force, have sequenced all of the DNA in a series of breast and colorectal cancers. One of our postdocs is coming back to do head and neck soon, and so we're hoping to reproduce some of this. But you'll go into a room with DNA sequencing machines going on all the way down a row and just sequencing night and day to gather this kind of data. And basically, the slides show what he calls peaks and hills of the percentage of the cases that have a mutation in a specific gene. And it's pretty much what was predicted years ago. There are maybe 10 to 15 major mutations in these cancer groups. But then there's this large group of targets that have lesser numbers or lesser percentages of mutations, but you can see that they also cluster in these little hills. So does that make sense of what we're seeing in the high throughput microarray that was on the last slide? Is it this sort of thing, or is there even more to it than that? Well, one of the things that we know now is that a major method or mechanism of genetic alteration is epigenetic, in this case mostly promoter hypermethylation, that turns off some genes from their activity without a mutation being present. And we'll hear a talk that focuses on that end of things. And then in the end, this was as close as I could get to the slide that Jenny Rubin-Grandis showed this morning. We get into these pathways, these major kinds of mappings, which are just our attempt to make some sense of the intense interweaving of alterations and the ramifications downstream, upstream control markers, and so on. And you get this impression, at least I do, standing back and looking at different people's pathways, that somehow they all do interweave in a very complicated manner that we just begin to understand. One thing does affect another somehow, and yet we need to make some sense of that as we try to start to do this molecular targeting and specific targeted therapy for patients. If we turn down one dial, do we turn up another, and do we need to block 3 different receptors or pathways or parts of a pathway at once? What are the end arounds that a cell can do to avoid the targeted therapy that we might bring to bear? Why is it that EGFR inhibition is not working as well as we want it to work? And so we're going to hear from Carter and Waz talking about some pathways, but others as well, as we think about how our individual genetic alteration information fits together into a physiologic gemisch that is much more complex than perhaps our tools, even as sophisticated as they now appear to us, can give us information about. So with that, by way of introduction, our first speaker is Bhuv Singh, clinician scientist at Memorial Sloan Kettering, and he's going to talk about the identification of targetable genetic events in head and neck cancer.
As usual, Wayne has stated the problem and all the issues that are facing us right now. Makes my job quite easy. So I'm going to give you more of a philosophical talk rather than a data-filled talk, and I'll use our data to kind of highlight what I think is necessary or is required to get to that next level. So what is the goal of our research in order to establish what that next level is? And certainly we want to identify these genetic events, but actually what we want to do is use those events, use them as predictors or even targets for treatment and, that ultimately lead to less morbidity and hopefully better outcome in the patients that we're actually treating. And in the past, it was quite difficult to try and find out what was abnormal. And the random approach was rampant, and P53 came out, so everybody looked at P53. Whatever was a gene of fashion, you looked at, because it was something that was feasible and doable. And certainly when PCR came out, you could start to look at a little broader array of abnormalities, but certainly nothing at a global level, just because the technology was not allowing a rapid throughput. And as Wayne just pointed out, there's a couple of seminal events that have kind of defined what's going on. And the Genome Project completion, in my mind, and probably most scientists' minds, is probably the most important contribution scientifically that's out there. And it changed it from a how many targets are there to establishing roughly how many targets there actually are. And then when you combine that with the newer technologies like Wayne was mentioning, where you can look at DNA level abnormalities, expression changes, changes in DNA that are normal DNA actually that change their functions, and so on down the line, epigenetics and sequencing, as was mentioned. Our ability to detect these changes became quite easy. In fact, what you do now to do array studies, you take a piece of tumor, send it to your core facility, and they give you back your data set for any of these things that are listed up here. So it's very easy to get these events, and the reality is we know a lot of genetic events in head and neck cancer, and we can actually begin to use some of these events to establish which patients are going to do well and which patients are not going to do well. And biologic parameters such as chemoresponsiveness, and as we've heard about a lot at this meeting, the papilloma status, as independent predictors can tell you who's going to do well and who's going to do poorly. Very strong prognostic factors. Genetic parameters, especially when you combine them at a global level of the total package of changes that are present, whether you do it at the DNA level or the expression level, or even panels of genes, you can predict who potentially will do well and who potentially will do poorly. But when you take this to that ultimate goal, which is to try and figure out if you can target these events and improve outcome, you don't get it. And I think the trials that are out there that target P53, the trials that are out there that target EGFR or some of the other receptor-based or membrane-based targets, they show very mixed results. And the reality is none of them actually show anything that's usable as a single agent. And so in spite of knowing this whole array of genetic abnormalities, we have not made any advances. And the reality is when you look at head and neck cancer, you have to understand that it's a progressive event. And as Wayne showed you, this progression can be seen often histologically or not, but certainly what's happening is the number of genetic events that it's in a cell is increasing as you go from a normal cell to a tumor cell. And prediction is there's probably somewhere in the range of 10,000 genetic events that lead to, or that are accumulating when a cancer develops. So any cancer probably, and specifically head and neck cancer. Now, a lot of these may not be in genes, a lot of these may be in promoters, they may be in nonsense areas within the genome, but that damage is accumulating. It is not a smart or targeted abnormality that occurs in these cells. Often it's a random array of abnormalities, and the ones that give you survival advantages are selected for. And so what you end up with is a circle that looks like this with a whole lot of events. And what the problem is that probably only a handful of those events are important biologically or even targetable for a therapeutic benefit. And so the challenge is not identifying the genetic events. We can certainly do that. The challenge actually is identifying the important events from the background of noise and random accumulation that has nothing to do with the actual cancer process or has nothing to do with cancer therapy. And the real issue is, how do we do that? So sometimes you get a home run right off the bat. You could do your kinome sequencing, which is what we did in collaboration with Harold Varmus at Memorial, and you can find mutations like we found in EGFR. And really interestingly, if you treat these patients with EGFR agents, which are already established and out there, what you find is if you have the mutation, you basically have a 100% response to the drug. That's a home run, and it's such a significant change that it actually now is part of routine evaluation of non-small cell lung cancers that come into our institution, and that initial screening, just like you would do a biopsy, guides your next level of therapy. And it's so important to understand how dramatic the results are. I think the best example is a patient that we've initially treated had bilateral lung disease, basically from top to bottom. Started treatment on Monday. On that Monday, she could not get out of a chair. She needed oxygen to maintain her oxygen levels. And by Friday, she was off oxygen. She can go to her local store, she can get her groceries and come back again. 5 days of therapy on that single agent. So this is a biologically relevant event. And this is what we're looking for when we try and find the important events in a cancer. And it would be great if this was present in head and neck cancer, and there's a couple of studies that have suggested that there may be some EGFR mutation, but for the most part, the critical mutations, the ones that are present in lung cancer, are not present in head and neck cancer. And to be quite honest, it's not a surprise because they don't occur in squamous cell cancers of the lung either. They only occur in adenocarcinomas, and specifically in bronchoalveolar carcinomas, more prone to in non-smokers than in smokers. And so the challenge really is to take your initial set of analyses, and I'll give you a brief snippet of what we've done within our own laboratory, of finding events, either it's chromosomal or gene expression based, and then doing the functional assays that actually validate those events. They validate them initially as targets and initially as the right gene in a specific abnormality and subsequently as targets. And so if you look at head and neck cancers, there's hundreds of events, and how do you pick one? And I'll tell you how we picked the event that we work on. We work on amplification at 3Q. Well, the first thing is it was pretty common in head and neck cancer, but even more common in many other different tumor systems, lung and other squamous cell cancers being present in about 50% of the cases. Amplifications are also tempting because in general, amplified DNA is unstable. And so the cell has to expend energy to maintain that amplified DNA segment. And the only way a cell's going to waste energy to maintain something is if it's getting a lot more back. And so we focus on this amplification because of its frequency and because of the fact that when we looked at it, we pretty much exclusively found it in heavy smokers, high-level amplification anyway. And we felt that there's probably something important giving the cell an advantage to survive. And that advantage, if you take it away, may give you an opportunity to target and make a therapeutic angle to that genetic abnormality. To establish it as an event, you need to do a lot of things. And even with all of this, it is not necessarily accepted. So you need to show that it's an oncogene. You need to show that there is oncogene addiction to the gene. You need to show that it transforms, and we've done it in cells, and we've also made transgenic mice and shown that in a mouse model, it actually makes tumors as well. And so this kind of tells you, okay, so it's important to the cancer process, and yet it still doesn't tell you if it's a targetable event. P53 is important to the cancer process, but all of the work that's been done, so far at least, suggests that it's probably not a targetable event. It's probably something that occurs early, and other things can compensate for it over time. So what you then go on to do is a whole series of experiments to define what the gene is doing and begin to understand the processes that it may be playing a role with. And I'm not going to go over the data, but we did in silico analysis, and what we learned is that there's actually other genes in this family, none of which were known, which we call, the gene that we found is we call SCRO, and the other genes are SCRO 2, 3, 4, and 5. And when we looked at these, we found that there isn't a head and neck cancer that doesn't have one or the other member of SCRO abnormal. In fact, there isn't a lung squamous cell cancer or a cervical cancer, and a high percentage of gliomas and a high percentage of ovarian cancers and many other tumor systems that we've looked at have one or another family member abnormal, and we focus then on how the gene actually works. And I'm not going to go into the biochemistry, but through reconstitution using purified proteins, what we've shown is that this gene, and the entire family for that matter, is a part of a process called methylation, which basically is a regulator of protein ubiquination. So downstream of this gene family is almost every protein that's important to human cancers, whether it's cyclin D1 or the COXes or the STATs, because almost all of them are regulated through ubiquination. And so what we've come down to is our thoughts being that everyone's been focused on how genes are made, and you make more genes, But if you have a really bad mutation in a gene that changes the gene quite a bit, it will be targeted, it will be ubiquinated, and it will be destroyed. So in order to maintain enough levels of that gene, you actually have to affect the destruction machinery at the same time as you affect the transcription machinery to maintain levels of that gene. And what we're thinking now is that this may be one method by which you can globally affect transcription, rather ubiquination, to allow you to maintain critical events within a cell. We've gone down to make models. We've made, as I mentioned to you, a transgenic mouse model. It's a conditional transgenic mouse model, and it does form tumors, but only in facilitated backgrounds. We've made a knockout mouse for our SCRO gene. We've now begun to make the knockouts for the other 4, 2 of which are almost ready. It's a really complex knockout background because there are 5 genes and they compensate for each other, and the phenotypes are actually driven by the compensation. So we've actually gone down to Drosophila modeling because there's only 3 of these genes in the Drosophila. We've actually made all 3 of the Drosophila knockouts. We're in the process of phenotyping, and we've also collaborated and done crystal structure. So why go through all of this? Well, I think this is what's actually required to understand what a gene is doing. And the reason I bring it up is that although you can now identify genetic events, the challenge is to identify what those events mean and how the genes are actually functioning. And so although it looks like on the surface we've made great advances and this may lead to therapeutic enhancements, the reality is that each of the events that you're looking at will require, like this, 2 or 3 years worth of work to delineate if they're important or not. Most of them are going to turn out to be not important. And so the hurdle is still there in terms of defining what are the critical events, and the hurdle is still there for the technology to catch up to. And the technology is catching up. I'll show you that within our own system, we can do a targeted screen. So we took a virus, we put in an RNAi against their gene, and treat it with a control virus versus the SCRO targeted RNAi and inappropriate cell lines which carry the amplification. The tumor is actually resolved. It's really great, but it can never actually come to clinical use. And so the second line of technology, which we're now concentrating on, is the ability to screen large pools of small molecules. So our institution has a pool of 300,000 small molecule inhibitors. We're in the process of setting up a screen. It's a 3-day process, and we'll know which molecules may actually directly target this gene. And yet even that process is slow, and it's still in development, and there is a lot of flaws, and we have to wait to get enhancements so that we can actually find drugs for each of the events that we find critical to the process. So the requirements are far from met. The requirements certainly are identification of the abnormalities, and at present, with the technology that's out there, we can do it. It's expensive, but it can be done. And I can challenge you to try and find a gene that isn't abnormal in head and neck cancer, because if you look at the literature, almost everything is reported in some percentage of tumors. So the requirement really is the assessment of the biologic significance of all of these things that have been identified. And that is indeed the challenge. We cannot do that at present in a high throughput manner. And this is where a lot of attention needs to be put in the future so we can weed out the background effects and identify the targets. And we need to get beyond our current approach. Our current approach is there's a drug out there, let's go try it. It looked good in cell lines, so it's probably going to work in tumors. But that clearly has failed because the vast majority of new compounds that are tried at the phase 1 level or even phase 2 never come to clinical use. And the only one that's at present for head and neck cancer that's used is part of a combination, and I'm not sure if it's going to be here 5 years from now. Although you see that one of the biggest booths in the drug guys area is that compound. I think the future is not here yet. I think the technology is not adequate yet, but I think it will be coming, and it'll be coming in the near future. And it's going to come from people in the audience, not only in the front row, but throughout. So with that, I thank you very much for your attention.
Thanks, Dr. Singh. I think we'll just hold questions and try to get the panel back up and talk about some things. Think about questions and write them down so that you don't lose them. I want to know more about molecular addiction. So we'll get to that. Carter Van Was, MD, PhD, and intramural researcher at NIH in Bethesda, is going to talk to us about pathways. And perhaps we'll go from how we identify individual targets to how they interact with each other from Carter. Thanks.
Wayne, thank you very much for the invitation to speak today. I have cooperative research and development agreements through NIH with the 3 companies listed, but I will not be discussing any of the work related to that today. So what I'd like to speak about today is we've all learned a lot about the genetic mechanisms of gene activation or inactivation through mutation, deletion, amplification, and more recently epigenetic mechanisms. And we also recognize through the work on epidermal growth factor receptor that signaling itself can play an important role in regulation of genes that may not themselves be mutated. And so my focus today is going to be to talk about mainly the regulation of genetic expression through signaling that results in activation of transcription factors, and what we can learn from array data to try and figure out how this works in cells, and maybe ways in which we can either use those pathways components as markers or as targets for therapy. So to begin with, I'd like to just remind you that carcinogenesis and the development of head and neck cancer involves a complex but a very stereotyped set of pathologic changes that we see in all cancers, and those include changes both in the cell phenotype and the environment, and these include increased proliferation of the epithelia, decreased programmed cell death, and in addition to that, which is due to the program that's inherent in these cells as they migrate up through the epithelia. It also is associated with a decreased sensitivity to therapy-induced apoptosis. And there's also increased migration, and in the case of cancer cells, invasion below the basement membrane. And the host response to this malignant transformation includes a number of changes, including increased angiogenesis and complex alterations in inflammation and immunity. So how do you explain all of these phenotypic changes? Is it, even if there are just a handful or a half a dozen or a dozen mutations that are critical for cancer development, how do we get to this kind of complex set of changes in most cases? And although there are differences in these features that are associated with malignant progression and differences in pathogenicity in response to therapies and clinical outcome, many of these same changes are observed in tumors that are less aggressive and very aggressive. So, so just to remind you about, and to remind myself about how we think about signaling, these kinds of broad changes in cell phenotype and the environment in homeostasis before cells become carcinogenic, or cancerogenic, can be regulated by changes in several levels. This includes signaling, gene transcription, and protein expression. And signaling occurs via cascades of factors, receptors, and kinase proteins. And this can lead in turn to signal activation of transcription factors that regulate the transcription of genes or DNA to messenger RNA. And these messenger RNAs in turn are translated to diverse proteins that regulate the cell phenotype environment. And this includes many of the types of changes that we observe in cancer and in normal responses to various things such as injury and wound healing. Many of the same types of genes are altered in both cases, including cells involved in, or genes involved in proliferation, cell survival, cell adhesion, and inflammation and growth. So the importance of this is that the proteins involved in these different steps, signaling, gene transcription, and protein expression, may provide a feasible number of key targets for drug therapy. And even if there are various alterations along these pathways that result in their activation, it may be that as we get closer down to the level of transcription, we have a handful of these, we may be able to develop tailored therapies for specific types of genetic alterations that ultimately lead to activation of these pathways and their stereotyped gene programs. So to begin to do this, we've done work with gene expression profiling in the past to look at specific pathways that are involved in regulation of gene expression profiling in murine animal models. And more recently, we've worked looking at human head and neck squamous cell carcinoma lines, comparing them to keratinocytes to try and explore the types of genes that are altered in a global fashion, and also to using cDNA microarrays that cover most of the genome, and also to begin to look at some of the known and lesser known signal transcription factors that are important in head and neck cancer. And this involves the process of analyzing this data and identifying a subset of the genes, of 24,000 genes in this particular case, that are altered or differentially expressed, and then performing a cluster analysis to see how these cluster together or in subsets, and then to take a look at all of these different clusters, and then to apply a high-throughput bioinformatic analysis to analyze the promoter or non-coding region of the gene that regulates its expression to identify binding sequences for signal-activated transcription factors. So taking this kind of data, genome-wide profiling reveals hundreds of genes that are differentially expressed either by most head and neck cancers or subsets of head and neck cancers when you compare them as a group to keratinocytes. And these include many that we would expect and also some that we don't expect that are involved or don't understand their role, but many that are involved in the kinds of changes that we see in cancer, including genomic stability, signaling, transcription, cell proliferation, survival, migration, inflammation, and angiogenesis. And just to pluck out some of these that are present in some of these either clusters, we have genes involved in DNA genomic stability in this cluster here. In this cluster here, those involved in inflammation, angiogenesis, signaling, and inhibition of apoptosis. And then down here, some of the ones you may recognize involved in inflammation, cell cycle progression, and adhesion. And then also downregulated genes that are we also recognize as being downregulated in many head and neck cancers, such as tumor suppressor P53 and P21, which regulates the cell cycle progression as well. So, in addition to these specific genes, we see that many of them cluster together, defining either heterogeneous clusters in which there's a difference between this group of cancers and the group of cancers on the left and the group of cancers on the right, or those that have more common expression in most of the cancer cells. So the way we've approached this is to do a high-throughput analysis of the promoter of genes, and we took this approach based on some earlier work we had done in which we noticed that many of the proteins and genes that were differentially expressed in head and neck cancer, if one looked at their promoter region, you could see that there was a enrichment for binding sites for certain signal-activated transcription factors, and these included P53 itself, but also a number of ones you may or may not be familiar with, such as nuclear factor kappa B, AP1, activator protein 1, EGR1, and STAT3, which you've heard about earlier today. And if you look at the promoter region and 1,000 base pairs upstream of the coding region of all of these genes, you can see a collection of various combinations of these factors which presumably can contribute to their differential expression in different settings, either alone or together among these pathways. So in order to analyze this in a more quantitative way, we looked to see whether there was an association between this distribution and enrichment for these binding sites in certain clusters of genes that were observed upregulated in these head and neck squamous cell carcinomas. And for example, you can see that this cluster A, which is expressed in about half of the cell lines here, is highly represented by p53 binding sites, but a lower level of NF-kappa B binding sites when you compare that particular subset. If you look at the other subset here, which we called cluster B, you can see that in that subset there's a low level of p53 binding sites, but a relatively high level of NF-kappa B binding sites. And similarly, for a number of the other transcription factors, you can see variable levels of increase in the representation of those binding sites over what would be predicted from the total vertebrate genome. And as you can see here also, just looking at the total vertebrate genome representation, that some of these transcription factors like p53 are present in about, at least predicted sites for them are present in about 1/3 of the genes in the genome, and NF-kappa B in about 50%, and for this EGR1 transcription factor also in the same range. But for example, for a number of these clusters, there's an increased representation of NF-kappa B or EGR1 or STAT3 or AP1 in certain clusters. So taking a look at cluster B, which is the one that has cells involved in cell survival and angiogenesis, these are associated with enrichment for NF-kappa B binding sites as well as STAT3, but a relative decrease in those or lack of p53 related sites. And interestingly, when we perform an analysis of the extent of transcription factor NF-kappa B activation in these cell lines compared to keratinocytes, in that particular gene cluster, there's an increase in NF-kappa B p65 DNA binding. And if you then look at wild type p53 expression, or this was a wild type p53 subset by sequencing, there's a relative decrease in expression of p53 compared to keratinocytes. And you can see that also down here by Western blot compared to those that express mutant p53, which is overexpressed at the protein level in most of those. So this suggested that there was an inverse relationship potentially between NF-kappa B activation and p53 expression and function in these tumor cells, even though they had wild type p53. And we explored this in tumor tissue and found that in addition to what we observed in cell lines, this cluster of genes and some of the expression of NF-kappa B in its activated form often was reciprocally related to low expression of p53, or conversely high p53 was related to reduced immunostaining for NF-kappa B. And together with that, a number of NF-kappa B related genes and their proteins immunostained together with NF-kappa B but not p53, and similarly were downregulated when there was high expression of p53. And if we look at a whole series of 24 tumors, you can see that there is a relative relationship between high NF-kappa B staining and low p53 staining, and conversely those tumors that have higher p53 tend to have lower NF-kappa B staining. So this suggested a hypothesis that some common mechanism that is involved in NF-kappa B and perhaps STAT activation, which is this other pro-survival pathway that Jennifer Grandis talks about, and p53 inactivation are linked together in this subset of tumors, promoting their survival. And this can easily be understood in terms of the expression of a number of pro-survival genes that are regulated by these factors, and the decrease in relative expression of a number of genes and proteins that are cause cell death, pushing this towards the left and promoting cell survival. So how do we examine this question and begin to work upstream and understand what's happening here? Well, one of the observations that was made in our lab that we pursued was that we had noted that there was a compound that had been identified. It's actually the anti-inflammatory and malarial drug quinacrine that had been observed in renal cell cancers to inhibit NF-kappa B while increasing the expression of wild-type p53. So we did a similar experiment in head and neck cancers and found that when we inhibited when we added quinacrine, that there was inhibition of NF-kappa B gene reporter activity with a reciprocal increase in these cells which had very low p53 expression. There was an increase in p53 with time and with its target gene p21. So this quinacrine-induced cell death, which you can see here from no treatment compared to the cells treated with quinacrine here on the right, is partially dependent on p53 because when we treat the cells, pre-treat them with a p53 inhibitor, this effect is attenuated as seen there. And the clinical relevance of this may be that quinacrine also enhances cisplatin sensitivity over what you see with either quinacrine or cisplatin alone, suggesting that this shift in the cell's propensity for apoptosis may be enhanced by such an approach. So this just illustrates that idea that something upstream of NF-kappa B can be inhibited by quinacrine, and this can result in also an increase in p53 expression and a shift in the cells towards cell death. So I haven't given up on p53 yet, and although I was quite reluctant to get into the field, I think this provides some evidence that there's not only a p53 to be upregulated in these cells that seem to lack its expression that are wild type, but can also be done by a drug that already exists and is approved for treatment of systemic lupus. So cluster A, this is the other subset. And so what is going on in this subset? And this was quite puzzling because this subset showed a relatively higher number of p53 binding sites in the promoter of its genes and a relatively lesser level of NF-kappa B in some of these other pro-survival signaling pathways. And because p53 was mutated in this subset of cells at the sequence level, we weren't sure whether the p53 was functioning or if something else was keeping the cells from dying in this subset. And so, as you know, p53 is now known to have several different family members, some of which, including p63, promote cell survival, like NF-kappa B, and those which also promote apoptosis, like p53, called p73. And we noticed that together with p53 mutant overexpression in these cells, that there was increased delta NP63 and increased p73, while there's a relative decrease in the level of NF-kappa B activity. So this suggested a model in which potentially p63, which is anti-apoptotic, could be serving some of the anti-apoptotic role to shift the cells towards a pro-survival setting. And some work had been published recently by Dr. Rocco's group showing that, in fact, delta NP63 binds up the p73 and can inhibit its ability to express pro-apoptotic genes, potentially shifting what NF-kappa B instead is expressed in these cells towards promoting survival. So that's our current model of thinking about this subset. And we've recently published some work indicating that p63 may form a novel interaction with the NF-kappa B pathway by forming a complex with a member of the family called CREL. And you can see here that delta NP63 and CREL co-localize in the nucleus of head and neck cancer cells, and that when you co-immunoprecipitate with using a CREL antibody, you can see on Western blot that delta NP63 is detectable. And when one does a CHIP assay, which is an assay for the binding of a delta NP63 to the promoter region of the p21 gene, you can see that along with that there's an increase in CREL binding as well as the increase in p63 binding, which is expected. And to see that there's a functional effect of this, if we transfect in CREL into these cells, and this work was done by Wendy Weinberg's group in the FDA with us, you can see that there's an inhibition of p21, which suggests that delta NP63 and CREL contribute to the inactivation of p21 in these tumors, and conversely that when you knock down CREL, there's an increase in p21 expression along with the growth arrest that's observed. So this suggests this model in which, in addition to P63's role in inactivating P73, there's also an interaction with CREL, which affects the expression of genes such as P21, shunting the pathway towards from growth arrest to proliferation. So I'll just wrap up by saying that we and others have begun to fill in the pathways that converge from the cell surface with the ligands and receptors to the kinases that are involved in these pathways, and now some evidence that these transcription factors are prevalent in the promoter region of many of the genes that we observe overexpressed in these tumors. And we can begin to think about these limited number of changes that, and targets that are down in this region here that we can use to affect global expression of many genes that are changed together, perhaps by a few alterations that occur up here and here that make it difficult to identify and treat many of these tumors because we don't know in any given tumor what the specific mutation might be. So in conclusion, we've found a set of signal-activated transcription factors, the ones listed here, that play a major role in activation or inactivation of gene programs important in the cancer phenotype. Activation of NF-kappa B and probably STAT3 is linked to repression of wild-type P53, while inversely linked with mutation status of P53 and expression of P63 and P73, revealing two important paths to development of head and neck cancer. In one subset, this activation of NF-kappa B is linked to inactivation of wild-type P53, and this subset has previously been somewhat paradoxically associated with greater resistance to chemoradiation therapy, and NF-kappa B is a known resistance gene to chemoradiation. And quinacrine is a drug that's currently available for study. It inhibits NF-kappa B, and it can reactivate deficient P53 in this subset in vitro, providing a candidate that can be studied for this. And lastly, the subset with mutant P53 that often overexpresses another family member, delta N P63, which has been shown to form a complex to inhibit apoptosis by inactivation of P53 and promote growth by forming a novel complex with the NF-kappa B family to inhibit P21. So drugs targeting signal activation of this pathway, along with inhibitors of NF-kappa B and CREL, may provide some new targets or combinations for therapy in this subset. So I'd like to just acknowledge all the people who have worked in the lab over the last several years on this project, as well as Wendy Weinberg's group that did the work in normal keratinocytes, in the collaborative paper that we published recently, and support by NIDCD for all the work. Thank you.
Thanks, Dr. Van Was. The next on your agenda is Andre Carvalho. Andre was a postdoc in the Califano lab at Hopkins and became our expert on hypermethylation, identification of markers, and now has returned to his native Brazil. I don't know what he's done since he went back, but we're going to hear.
Good afternoon. I'd like to thank Dr. Wayne for the invitation to talk about epigenetics and methylation as a mechanism for development on head and neck squamous cell carcinoma. So I have nothing to disclose for this presentation. And just going quickly about the definition of epigenetics. So epigenetics refers to changes in gene expression that are stable between cell divisions and sometimes between generations, but do not involve changes in underlying DNA sequence of the organism. So basically what we have an addition of a methyl radical in the cytosine, so we became to have the methyl cytosine. And what is happening is that this, when we look at the inactivation of cancer genes for genetic or epigenetic events, and need to have two events, and one of these in any of the arms can be a DNA methylation for a specific gene. It's have been known that the global methylation comes with age, and this can lead to hyperexpression of oncogenes, but what we are mainly talking about is hypermethylation that is gene-specific and will actually gene silence. And when it comes to a tumor suppressor gene, we can develop cancer. Basically, when we have here the promoter region that's unmethylated, then we have the RNA expression. But then when you have a methylation CpG island in the promoter region, you cannot have the RNA expression anymore. There is mechanisms to do that, so addition of acetylation and the methylcytosine would lead to condense chromatin, and then this will lead to gene inactivation. As I told before, to finish this topic, is that we can have DNMTs to do de novo methylation, DNMTs 3A and B to maintain the methylation. When you have the cell division, you're going to keep the methylation within the cell division, and this methylation for different mechanisms will stop transcription. Nowadays have been discussed that siRNA also plays a role in methylation. You can have establishment by action of siRNA, and also the maintenance of hypermethylation when it's done methylation. There's a number of current methods for student DNA methylation. I'm going to talk for the most common one. That is when you bisulfate treated the DNA, if you have a cytosine that's methylated, when in the bisulfate treatment, it keeps as cytosine instead of when you have a cytosine that's not methylated, you have uracil, and then you have a different products after treatment in the PCR due to the methylation or not. After that, you can basically then sequence, and then you can have an unmethylated sequence with the TG and a methylated sample when you see the CG. We can do this in a high throughput. We can design probes to look at the real-time quantitative MC PCR. And here, example, we have a standard curve in pink, and then we have for beta-actin our 3 samples showing the DNA is methylated. Here, one is highly methylated, one has less quantity of methylation, and one sample we can see that's unmethylated. How we select genes for saying that that gene is methylated and this is the reason for gene silence, one of the ways most usually at the treatment with the demethylated agents, what it do is we try to find a cell line that for a specific gene that we are looking for has no expression. For instance, for FADU cell line has no expression for this gene, O13 has no expression for CA3, and then we treated that cell line with 5-ASA cytosine. And then we have in the day 0 before the treatment, no expression for that particular gene. And then after treatment, we have the re-expression of that gene. And these probably say that methylation was the reason that that gene was silenced. Here we have the same example for another gene. The confirmation that it was hypoexpressed in 75% of the tumor cells, the squamous cells carcinoma that were tested for the 2 genes. But then when we went to the clone of the sequence to see of the CpG island, we can see that the O13 that has no expression for that particular genes has all the promoter region methylated. The O28 has also promoter methylation, but you would see expression and the RT-PCR, and the O13 after 5-ASA treatment still keeps with the promoter methylation but begins to expression. So sometimes only to do the RT-PCR after 5-ASA cytosine do not confer to you that that gene was inactivated by hypermethylation. And here, the other gene that we tried with the FADU cell line, we can see just few hypermethylated CG islands in the promoter region. This cell line would not express. This cell line will express, and we don't see that places with this hypermethylation, and after the treatment with 5-ASA, we don't see anymore that hypermethylation promoter regions, and we begin to have the normal. My question here is that, is this enough CG islands to be hypermethylation and inactivate the gene? It's kind of, sometimes we are not sure about that. How do you select the genes that we want to see if it's hypermethylation? Another way in a high throughput is to do the 5-ASA in a microarray, the cell lines, and then you become with a lot of genes that are upregulated after when you compare the cell line expression before and after the treatment, and then you can choose your candidates. This is a more sophisticated way to do that. Patrick Ha was involved on this study that you compare the results with your cell lines with the results that came when you compare the expression of head and neck cell line, squamous cell tumors with normal samples, and then every gene that's downregulated may be related to hypermethylation. If you compare with that results that came from the 5-ASA, you came with some common genes, and these must be your more active candidate genes. From this, we came with the DCC gene that we look for to see if DCC hypermethylation was related to carcinogenesis. We look at 632 tumors, and 143 tumors, and 76% of them has hypermethylation of DCC. And when we look for the saliva from control individuals that do not have head and neck cancer, only 1.3% of 632 controls has hypermethylation. So it means that hypermethylation for DCC is specific for patients with head and neck cancer. We also look, when we look for only the patient with head and neck cancer, that the presence of DCC hypermethylation was related to alcohol consumption and also with race, where the African American has less hypermethylation of DCC than the Caucasians. We then look at, for the role in the carcinogenesis, we have the immunohistochemistry showing no expression of DCC or the expression of DCC, and we could look that in the cases that there is negative for immunohistochemistry, 91% of them has promoter hypermethylation for DCC. We also went further and looked for the transfection of DCC in a cell line that do not express DCC beforehand. So we did our CMV vector transcription and transfection. There is no expression, but when we put back the DCC wild type and that transfection, the cell line re-expresses DCC. When we went to our colony Fox assay, in the mock transfection, we still see proliferation from the cell line, but when we put DCC wild type back there, there is no proliferation or very few proliferation in the cell lines. So this is a more strong evidence that hypermethylated in DCC promoter region is really related to gene expression and gene silence. But yet what I want to comment is that sometimes we can't find that link what we saw or what we detect of hypermethylated and gene silencing. But yet, when you have a gene that's only hypermethylated in your cases, and it's specific for your cases, still there is a role for a potential tumor marker. So, and this is what I'm more working nowadays, is how to use hypermethylation as potential marker for diagnosis, surveillance, and prognosis. One of the things that allow us to do more with hypermethylation, that you only need the DNA, so you can get for any body fluids as saliva, sputum, serum, urine, and go on. One thing that we need to know also is that there is a compartment-specific methylation when we are detecting methylation. For instance, for the Epigenetics, when we look at the tumors, there are 83% of the tumors that present hypermethylation. But when we look at the controls, taking in account saliva and serum, For saliva, there are few cases, only 10%, and low levels of hypermethylation. But when you look at the serum, almost 1/3 of the normal individuals who have DAP kinase hypermethylation detected on serum. So DAP kinase might be a good marker for saliva, but not for serum. When you look for HAR-beta-2, it go the other way around. 83% of the tumors has hypermethylation for HAR-beta-2 gene. 62% of the saliva control has this hypermethylation and almost the same level. So HAR-beta-2 would not be a good marker for saliva, but none of the controls would show HAR-beta-2 methylation on the serum. And you also have genes that, regardless of the compartment you're looking at, you're going to have a high number of normal controls that do have promoter hypermethylation detected. This is our panel that we try to develop to head and neck cancer diagnose in saliva. This is the plot for specificity and sensibility of head and neck cancer in saliva rinses, considering 175 cases and 450 controls. This is every plot is a different combination of the 25 genes that we try in this panel. We can see that if we want a panel with more specificity, we can have very low sensitivity. So what we get more, it's some panels that have around 35–40% of sensibility with over 90% of specificity. So the best panel it come out has 7 genes, and when we did that panel in our head and neck cancer patients looking for hypermethylation in any of those set genes that come out in the panel, these are the results for local control. Almost 90% of local control when you don't have your saliva pre-treatment methylated for that panel. In 5 years, around 6%, and after that, around 35% of local control when you have your saliva pre-treatment methylated for at least one of the set genes. And this is also stay in when you count for overall survival. When your saliva is unmethylated, your survival for those patients are much better than when the saliva is methylated. We also are looking for markers during the follow-up of the patients. So Hopkins has a prospective study going on, collecting the saliva and serum prospectively before treatment and after treatment in each time the patient comes for a follow-up. And the reason is that we are trying to look for markers that when you have still your patient with a normal clinical exam, maybe if your patient will recur, maybe your methylation saliva could be detected beforehand. We don't have a result on that yet, but there is a publication from a French group taking into account 90 patients, which 22 patients were follow-up and 6 genes were done. So I just want you to talk about this part here. They have 22 patients that they have saliva collected during follow-up. From the 17 patients that has no methylation, there is only 1 relapse. And for the 5 patients that was detected hypermethylation of the saliva during the follow-up, 5 of them have recurrence. So showing that hypermethylation has probably a big role in local control. Also, I just wanted to touch about the possibility of using those drugs that we use for demethylated as a treatment for the patient. This has been used in myelodysplastic syndromes. When you compare the use of a demethylated agent compared to supportive care, there is a better survival for the patients that was in use for the demethylated agent. And also there is some clinical phase 1 and 2 trials using histone acetylase inhibitors in some tumors. Here is all the tumors they are trying. As you can see, there is no head and neck tumor that have been trying these kind of drugs yet. for hypermethylation. As conclusion, I'd like to say that methylation plays an important role in head and neck squamous cell carcinoma carcinogenesis. Its detection might be useful for diagnosis, surveillance, and prognosis in head and neck squamous cell carcinoma patients. So even if, and I would like to emphasize this, you don't have a proof that hypermethylation really silences your gene, if your detection is specific for your patients, it still can be used as a tumor marker. And hypermethylation can be a target for molecular treatment. I'd like to thank the group that was in Joe's Califano lab when I was there doing my postdoc, and the group in São Paulo that I work with. Thank you so much.
Thanks, Dr. Carvalho. So our final speaker for the panel before we open to questions is Patrick Ha. Patrick is on the faculty at Hopkins. We wanted to have someone talking about something other than squamous cell carcinoma. We know that there are lots of other head and neck type tumors that we treat, and Patrick has been focusing his attention on adenoid cystic carcinoma. I guess for the molecular biologist, it may not make too much difference what cancer we're looking at, and yet we certainly are confounded by these very unusual tumors that we see a lot of in our clinical practice, and yet nobody is studying. The adenoid cystic cancer patient group is very active and very interested in what we can do for them as clinician scientists. And so Patrick's going to let us know some of what his progress has been in that regard.
Thank you, Wayne, for allowing me to participate in this wonderful panel. I'd like to start with an apology for all of you who are expecting a talk on paragangliomas and pheochromocytomas, but I made a strategic decision that I should talk about something I know something about. So I figured this would be a more worthwhile talk for all of you. But like Wayne said, my focus has been on adenoid cystic carcinoma, the salivary glands. And unlike many of the talks that came before that were very specific, I can only give a brief overview, and part of the problem is that there hasn't been that much done in adenoid cystic carcinoma. So that's one of the good things, is that I think there's a lot of work to be done. We can utilize all the technologies that everyone has been discussing. And the other good thing is that I can give you an overview in 20 minutes. So with that, let's get going. Okay, so I have no disclosures. John Connelly, who all of you know, had one of the most significant and largest experiences with head and neck malignancies. And even to Dr. Connelly, in one of the quotes that he published, he found that adenoid cystic carcinoma was one of the most deceptive and frustrating in clinical management. And I know that all of us who have taken care of these patients feel this way and understand what he's talking about. While local regional recurrence may be low, we have a good opportunity of trying to cure them locally. We know that we're going to fail distantly. We don't know in which patients, and we don't know how long they're going to survive with their metastases. So it's a very frustrating disease, I think, for clinicians, and it's difficult to know how to counsel patients and what to do with the rest of their lives. So we're really at a loss for what to tell them, both in terms of how we're going to treat them and manage them, and how we're going to counsel them and what to expect. So just for a brief overview, it's a relatively rare disease. Within the U.S., it's 1 to 2% of head and neck malignancies, so there's only maybe 500 to 700 cases per year. It happens generally middle age, a little bit more predilection for women than men. And there's really not any exposure or known exposure or genetic syndrome that leads one to adenoid cystic carcinoma development. And so oftentimes in other tumor models, we've hang our hat on these various patterns to try to see why exactly or what exactly are the genetic mechanisms that underlie this. So clearly this is probably a sporadic mutation. It may be consistent, but it's one that's probably not well described. And it does tend to affect patients in the minor salivary glands more than in the major salivary glands. In terms of the numbers, these are the 5, 10, and 15-year survival rates. And again, it's relatively unique in that we don't usually talk about 15-year survival rates in most cancers. So while the 5-year survival may be on par with other head and neck cancers that we're familiar with, 15 years out, about half your patients are still going to be alive. The other confounding thing is that they may be alive with disease. They have pulmonary metastases, but they're doing fine. They're completely clinically asymptomatic. We do know that the average time is probably about 36 months till they develop metastases distantly. But like this is from Spiro, his range was in 1 month to 19 years. So 19 years later, a patient may come back with a pulmonary metastasis from the adenoid cystic carcinoma that you thought you treated 19 years ago. The other interesting fact is that survival after metastasis, again, 5, 10, and 15-year survival, there's not many tumor models where you can look at these numbers and say, after you've developed distant metastases, 40% of patients are still going to be alive in 5 years. And if you look even further on down, it's very unusual. So one of the issues with treatment is that while there are many nuances to surgical and radiotherapy techniques, really we've gone maybe as far as we can so far in terms of surgery and radiotherapy. We have a pretty good chance of having local regional cure, or at least control. And now we're looking more for how do we control the systemic disease, looking at cytotoxic chemotherapy agents and more specifically look at molecular targeted therapy. And we've essentially just borrowed technology or treatment from other tumor models because there hasn't been very much done looking at the underlying mechanisms of adenoid cystic carcinoma. So this is a nice summary of the cytotoxic agents that have been tried in adenoid cystic carcinoma, and I'm going to highlight some of the problems, and one of the difficulties in studying this disease is that if you look, for example, the total number of patients, this is a meta-analysis looking at many different studies, you can see that the patient population is very small. Any one of these is, 13 at most, 43 patients, and that was over 5 or 6 different trials. The other problem is that response rate is something that's very difficult to understand or to categorize. Many times response may be defined as regression of disease, which is ideal. Stabilization of disease in adenoid cystic carcinoma may not mean anything because that patient, if you did nothing, might have stable disease to begin with. So you have to be careful when you look at the response rate. And so looking at the cytotoxic agents, it's really rather dismal. There's really not a whole lot of response, 2 out of 13, 0 out of 14. And so what we've looked at is trying to look at more molecularly targeted therapies that may not be as toxic, but may have hopefully an equivalent effect in these patients. And so that leads us to looking at what is the molecular biology that is underlying this and trying to identify the mechanism so that you can then target it in a rational fashion as opposed to just trying whatever is the new drug of the day. So going back to the basics, looking at cytogenetics of adenoid cystic carcinoma, these are sort of the common things that have been found. There's a lot of literature looking at sort of isolated case reports of this or that, but these are sort of the consistent things. 6q24, 2124 deletion, 12q1213 deletion, 20q13 gain. And these are sort of the putative genes that people have thought might be related to this, to the carcinogenesis. Now, none of these genes have actually been proven or consistently found to be either upregulated or downregulated in adenoid cystic carcinoma. And this last study here that Dr. Miskalek's group performed out of University of Virginia kind of highlights this in that, they surveyed 22 tumors, 22 normal, and they found an average of 3 deletions in any one of these tumors. And even then, there was no consensus area of deletion. So clearly sporadic and not consistent and very rare. So it has a very stable genome. So we're probably not going to find it through any cytogenetic approach. Looking at expression rates, another great avenue of technology that's come about. Again, looking at this, these are the types of genes that tend to be altered. Developmental genes tend to be upregulated. Apoptosis genes tend to be downregulated. Cell cycle, downregulated. Matrix metalloproteases, which is thought to be related to cell adhesion and metastasis, are upregulated. Perineural invasion genes tend to be upregulated. And then there's more specific genes looking at SOX4 and Wnt pathway genes that have also been sort of sporadically mentioned. And we'll touch base on a couple of those pathways. So one of the biggest hits, so to speak, for adenoid cystic carcinoma is c-KIT. Which is a tyrosine kinase inhibitor. We've heard a lot about tyrosine kinase inhibitors. And this is a proto-oncogene, and it's found most commonly in AML and GI stromal tumors. And so people did immunohistochemistry in adenoid cystic carcinoma, found that in a high percentage of cases, it was upregulated in like 90 to 95% of cases. So, great target. On the other hand, there was no mutations in exon 11 or 17, which is what was commonly found in the AML and GI stromal tumor cases. Nevertheless, it's as good a hit as we had, so why not move on with that? Imatinib, or Gleevec, which is a specific c-KIT inhibitor, was found to be very effective in CML and GI stromal tumors. And within our literature, there was a case report of 2 patients who were treated clearly off-label with imatinib, and they actually had regression of their disease to the point where they were unresectable initially and they became resectable. One patient received resection, and the other patient opted for no surgery, but did indeed have visible regression of their disease. Based on this, a formal multi-center trial was established, and they quickly enrolled 16 patients. Oops. And the problem was that there was no regression. None of these patients regressed. They had 9 instead of 16 patients with stable disease. And again, that's not really response if it's stable, at least in adenoid cystic carcinoma. And then 6 patients actually progressed, so they closed that trial due to a lack of response. This was also performed separately in a separate institution around the same time, and that also was closed due to a lack of response in their patient population. So looking at other tyrosine kinase inhibitors, EGFR, which you've heard a lot about, it's also been found to be overexpressed in up to 85% of adenoid cystic carcinomas. But that upregulation hasn't been as consistent as with c-KIT. But nevertheless, it's what we have. So Gefitinib and cetuximab have been tried in these patients. And again, stable disease 68%, stable disease in 50%, with no clear regression in either of these groups. So again, disappointing results. AEE788, which is a relatively new study coming out of MD Anderson, which is a dual EGFR and VEGFR inhibitor, was used in a mouse model. So this is one thing I wanted to highlight, was the use of these mouse models will, I think, become very useful, particularly in these rare tumors. But what they did was they took an ACC3 cell line, which is the only reliable known cell line in adenoid cystic carcinoma, implanted that into the parotid glands of mice, and treated them with paclitaxel and this combination drug. And actually, they did show good results. So it inhibited growth and prevented lung metastases in these mice. And as a corollary, they found their decrease in MMP9 and MMP2 expression which may be important in the mechanism of preventing metastases. So we'll talk a little bit more about potential uses of mouse models, particularly in these rare tumors, in a little bit. Going through their laundry list here, HER2, which is part of the EGFR family. This has had a variable rate of overexpression in adenoid cystic carcinoma. So that in the literature, it's somewhere between 15 and 75%. So what Glisson et al. Wanted to do is to try to figure out whether this would be a useful target. And they actually started a clinical trial, and actually this is of all salivary gland malignancies. 137 patient screen, only 17% actually had upregulation of HER2. So they were planning on giving trastuzumab, a HER2 inhibitor, but they actually decided that this trial was not worth it because 83% of their patients they had to turn away and say they couldn't deliver the medication. This was lapatinib, which is a different type of HER2 inhibitor, both with HER2 and EGFR. That was, again, 19 patients treated, no response, 15 with stable disease. So now we move on to maybe what you consider more isolated genes, and SOX4 is one that's relatively new, and part of the problem is that there's really not much known about it, except that it was upregulated in one of the large screens of adenoid cystic carcinoma using microarrays. It's expressed during development, loosely associated with various tumor types, and in this particular paper, again out of University of Virginia, the knockdown led to increased apoptosis in the ACC3 cell line. But again, there's nothing further been out about the true mechanism of this gene. So we move on to promoter hypermethylation, which Andre thankfully introduced to you all. And so it's something that I have particularly been interested in, specifically because of the relative genetic stability of adenoid cystic carcinoma. You tend to think more about epigenetic events or more regulatory events, and not necessarily gene mutation, deletion, or sequence alteration. So these various targets, 1433 sigma, MGMT, RAS, F1A, DAP kinase, P16 as well. These are all players that have been seen in head and neck squamous cell carcinoma, and they've been upregulated to methylated to more or less different degrees depending on the study. Some of our work we've done is to use the quantitative methylation-specific PCR that Andre alluded to, which adds the benefit of being very sensitive and very specific, because it adds an extra probe in there so that you're sure that you're actually querying the sequence that you were looking for. In addition, it gives you quantitative numbers so that if it's a very, very low level of methylation, then you may not think that that's as clonally specific. So we've actually screened 15 candidate tumor suppressor genes, either stuff that's been reported in the literature previously or just genes that we think may be interesting within adenoid cystic carcinoma. And interestingly, 9 out of 16 of these genes in our normal salivary gland tumors actually showed promoter hypermethylation, which is interesting because if these are truly tumor suppressor genes, you would think that their function ought to be the same in salivary gland tissue, but what we're learning is that salivary gland tissue is very different than squamous cell mucosa. And so really only RASF1A came out of the study as being significantly methylated, which is one of the targets that we're going to be pursuing further. So the limitations in what we've been trying to do has been that it's low incidence, so it's hard to accumulate a significant experience in any one institution, which leads to difficulty sometimes in obtaining tissue samples. And then the lack of cell lines, for example. There really is only one reliable cell line that individuals have published on. There are some other cell lines out there, but sometimes they don't necessarily recapitulate the same behavior as adenoid cystic carcinoma, either in mouse models or even in vitro. And then, of course, funding mechanisms have been an issue, and that's not specific to adenoid cystic carcinoma. But the ACCRF is a research foundation that has been started by Jeff and Marnie Kaufman. Marnie, I'm not violating any HIPAA rules, but she is a survivor of adenoid cystic carcinoma. She has a vested interest in this, and they've really brought the adenoid cystic carcinoma research community together to form a very collaborative environment, one where we can really share resources. And I think that's the way that you have to approach a disease like this. So one thing that our lab has also been very involved with is looking at xenograft models. And so, as most of you probably know, you implant the human tumor tissues directly into an immunodeficient mouse, either nudes or skids, doesn't really matter. And they will actually recapitulate the tumor behavior much better than cell lines. So you can actually grow these tumors, you can divide them and passage them into other mice. And so actually what it allows you to do is to amplify the amount of tissue that you have so that it's nearly infinite. And then it's an easier platform in which to derive cell lines, and that's something that we've been actively working on in our lab. The other aspect is that you can run mini clinical trials. So rather than trying to collect adenoid cystic carcinoma patients in multiple institutions where each one can contribute maybe 2 or 3 patients, you can actually bank these tumors, collect them in a central area, and then treat them essentially with combination drugs, different inhibitors or cytotoxics, maybe in combination. And it gives you a much better idea of what is happening biologically, and you can do it in the mouse before you actually bring it to humans. So overall, I hope I've talked to you about, explained sort of what the background is of adenoid cystic carcinoma and why we've had such a problem managing it. And I think that really one of the issues is that we don't really understand what is the underlying mechanism of this disease. We're quick to rush to borrow any technology from any other tumor model, but I think that really we have to go back and figure out the fundamental basis of this disease, and it may be that it's a very different pathway than any other tumor model that we've studied, and it's our hope that once we are able to find out a little bit more about this disease using all the tools that we have at our disposal, that we'll be able to get a better handle on treating this disease and treating our patients. Thank you.
So we have about 6 minutes. I'll ask the panelists to come back up, and if you have a question that you'd like to ask, come to the microphone, and we'll try and answer it. I'm curious about what you guys think. We've talked a lot about what happens within a tumor cell, and particularly when you think about the signaling pathways. Obviously, the signal has to come from somewhere. What do you think about what's happening in the larger tumor microenvironment, particularly interaction with stroma, but also interaction from tumor cell to tumor cell, and how does that interplay with what we can figure out when we look at cells sort of in isolation?
I think that's the less analyzed biology, if you will. Clearly, the stroma plays a big part, and the interactions between tumor cells play a big part in how a tumor behaves. Probably the best example is angiogenesis, where if you knock out a specific gene, IT3, you won't get angiogenesis. But if you were to take an IT3 knockout mouse and do a bone marrow transplant with wild-type bone marrow with normal IT3, it would reactivate the angiogenic signal, which has actually not even stromal cells. It's actually bone marrow cells that are leading or allowing for progression and tumor growth. I think the biggest problem is it's hard to study those interactions. It's hard to recapitulate them in a petri dish. And I guess you can co-culture with fibroblasts, but then how do you introduce everything else, and how do you decipher the signal from the noise if you're going to look at primary tissue? And so it's a harder series of experiments and probably something that's not easily accomplished, at least at present. So I think the answer is we have no idea.
I was afraid he was going to say that.
I could comment on that a little bit. I mean, there are many of these signaling pathways have negative regulatory feedback loops. And when one looks at some of the genes that are methylated, for example, one example is in the interleukin-6 pathway, where STAT is activated, there's a loss of SOX, which is the suppressor of cytokine signaling. So if there is IL-6 in the environment, either from the stroma or being made by the tumor cell itself, the absence of SOX in a subset of tumors that have that defect are more likely to be responsive and maybe more dependent or more driven by IL-6. So I think that's one example that we've teased out from some of the cell line studies. Obviously, a lot of that has to be validated in vivo, but those are the kinds of things. The loss of negative feedback, I think, is one of the key ways in which the genetic or epigenetic mechanisms influence the receptiveness or the increased responsiveness of these signaling pathways.
Yes.
As you know, tumor genomes are highly heterogeneous as a result of the massive genomic instability that occurs. When you're looking at your arrays, whether it's expression arrays or CGH arrays, you're looking at the average of the entire tumor. So when you're seeing a 10-fold elevation in expression, you may be seeing 10-fold in every cell or 100-fold in 10% of the cells. And if you're designing your targeted agent against a particular increase in expression or amplification, you're going to knock out 10% of the tumor and leave the rest behind to kill your patient. So the question is, nobody has, at this meeting, as far as I know, has mentioned heterogeneity, measuring heterogeneity, and I think that's a critical thing to start looking at in deciding who gets what therapy. Anybody want to comment on that?
You know, I missed part of the last part of the question. I couldn't quite hear. It was a little bit garbled from
Nobody's mentioned heterogeneity and how we target or think about heterogeneity when we're thinking about these methods that allow us to sort of analyze a tumor in toto. Certainly the methylation information that Andre showed talks about the percentage of cells within a tumor that have an alteration. Now that doesn't necessarily, actually I guess it's the percentage of the tumors in a population, though.
So I think it's a very valid point. I think the landmark experiments that highlight the importance of heterogeneity are probably Joan Massaguet's experiments, where he's taken a cell line, put it into a mouse, and been able to show different compartment of cells having metastasis to different organs. And then once you look at the profiles of those, he's been able to identify genes that are dysregulated in that subset of tumors that metastasize to an organ, and can recapitulate the metastasis in all the cells by introducing a handful of genes that are in there. But you know, what you're describing and what you're talking about is the real limitations that are present in tumor analysis. You can't do a cell-by-cell assay as yet. And then, you know, if you compound the fact that you can't actually detect everything in all the cells that are occurring, that may affect a single gene. And if you take that to the next level, some of the things may not be abnormals. They may be normals, such as SNPs or post-transcriptional changes in a protein. I think what you're pointing out is how complex these tumor systems are. It is not as simple as a hematogenous tumor where you have a single abnormality that drives the phenotype. It's a very complex phenotype, and may not be present in all the cells. That would explain the recurrences after chemotherapy, for example. You treat the cells, and most of them get killed off, and you have a clone that survives. Usually it's more aggressive if it survives the chemotherapy. Again, I think this is a technology issue, and it probably will be addressable in the future, but not at present.
What's this process that you referred to, Bhuva, of a 3-day setup to look for the small molecule screen?
This is something new that our institution just got. It's a biocomputer, which is a substantial biocomputer. and it can do screens the drugs over a 3-day period on either cell line or biochemical processes with fluorescent endpoints. So over a 3-day period, I can run the computer and screen 300,000 compounds.
So it's an in silico screen? Is it?
No, it's actually the endpoint is either fluorescent-based detection of a biochemical process or cellular survival. So it's actually a in vivo screen if you want to look at a cell line as in vivo, or a purified protein screen. And it's not just present at Memorial, it's present in a lot of different places. And a lot of different drug companies are actively involved in it.
Well, by my watch, our time is up, and so unless there's a pressing question, you can come up and ask the panelist. We're finished with this session, and we'll see you at the next one.